today I'm going to be here. Thank you. And today I'm here to present about domain-specific biochemistry problem solving in undergraduate students. So, to start off, we're investigating undergraduate student problem solving about threshold concepts in biochemistry. Now, we define a problem as a task that presents a challenge and cannot be solved automatically. In addition, problem solving is a decision-making process where a person is presented a task and the path to solving this task is uncertain. Now, some research questions. To what extent do students and experts use domain general, domain-specific knowledge when solving the problem? To what extent do students successfully solve problems? And what are the key similarities and differences among students and experts while solving problems? So, to study problem solving, we use the theoretical framework of domain-specific problem solving. And this framework of problem solving acknowledges that good problem solving requires a well-organized knowledge base and application of the knowledge of the problem at hand. In addition, we have a model that consists of specific and general problem solving, specific being knowledge that is exclusive to the topic, and general being knowledge that can be used in other domains. So we further categorize these knowledge types as declarative, procedural, and conditional. Declarative being content knowledge or factual knowledge, procedural being problem-solving processes, and conditional is when to apply these knowledge and processes for a given task. So we are investigating our research question by applying this framework, and we're also investigating whether there are interactions among these types of knowledge, as others have already hypothesized. So, to help answer our research question, we connect domain-specific problem solving to threshold concepts in biochemistry. And just a brief summary, a threshold concept is a concept when mastered represents a transformed and integrated understanding of a discipline, and without which a learner cannot progress. So that means that these concepts are crucial to the success of the discipline. And these concepts are transformative, integrative, bounded, troublesome, and irreversible. And there have already been threshold concepts identified in biochemistry, and today I'll be specifically focusing on the physical basis of non-covalent interactions, which is what is pictured there on the right. Now, to investigate our questions, we use this problem, Protein X problem. And students and experts are given this problem to complete. Shown here is a folded protein with phenylalanines in the middle. Students and experts were asked to predict the effects of protein folding and function for each amino acid substitution, glutamate and valine. You can see that on the bottom right of the depiction. We collected data from three populations: introductory biology students, which represent our beginning students, biochemistry students, our advanced students, and biology, chemistry, and biochemistry PhDs, our experts. Now, we performed lab interviews, which allowed students and experts to verbalize their problem solving. And then these interviews were video recorded and transcribed. From the interviews, we developed a code book where we coded individually and then compared the codes as a group in an iterative process. And the final outcome were codes that were categorized into our categories: procedural, declarative, and conditional knowledge types. Now, for some results. To answer the question, to what extent do students and experts use domain general and specific knowledge when solving the problem, we use the codes to create timelines. And our timelines show how experts and students solve a problem from start to finish. Now, we have 6 things on our timelines depicted in different colors. The first one is green. It represents accurate domain general problem solving. Next up is inaccurate domain general problem solving, green with black stripes, and purple is domain-specific problem solving, and then inaccurate domain-specific problem solving. Purple and green stripes together, as you see the second from the last, represent both domain-specific and general being coded at the same time. And just to be clear, these were codes that were both present at the same block of time in the transcripts and the interviews. When you see blanks in the timelines, they represent where no code was given at that moment, and this could be pauses or questions that the interviewee might have asked. So we're going to zoom in on 3 timelines here. Pamela in the very front is an expert, and Samantha and Emily down below are students. So right away you can note that experts and students use both domain general and domain specific knowledge to solve a problem. Let's see if I can point that out. Domain general and domain specific to solve a problem. But however, you can see that the experts, Pamela, are more accurate. They don't have the stripes and spend a significantly less amount of time on problem solving compared to students. Here are all the expert timelines shown in comparison. And you can see here, experts primarily use domain general problem solving to frame the question, followed by domain specific problem solving to answer the question. And once again, comparing all of them, experts are highly accurate and very quick to finish the problem. The quickest expert finished the problem in 3 minutes and 44 seconds, and the slowest was 9 minutes and 17 seconds. Now, comparing all student timelines, you can see again, students use both general and specific knowledge to solve the problem, but they primarily use domain general problem solving interspersed inside the problem solving steps as well. The fastest student, Paul, completed the problem in 11 minutes and 6 seconds, and this was still slower than the slowest expert interview. In the longest interview, Robert completed the question in 20 minutes and 30 seconds. As you can see here, there's quite a variance in student response. So, we wanted to understand the variation among students and which students reached a successful conclusion or answer. And that brings us to our next result, which is in order to investigate to what extent students successfully solved the problem, we applied a scoring rubric to define success among students. And here it is. I'll explain a little bit about it. Starting from the left, we can see the participants, all student participants here, followed by their level of study, advanced or beginning, and then the 10 criteria that we deemed represented success for a student. And those are, starting from the left, protein folding affects function, phenylalanine is nonpolar, glutamate is negatively charged, glutamate charge needs to be satisfied, glutamate mutant will fold differently, glutamate mutant would function differently, valine is nonpolar, phenylalanine and valine would interact through van der Waals, valine mutant would fold similarly, and finally, valine mutant would function similarly. The students were given a number 1 to represent a correct response for each criteria, and a number 0 to represent an insufficient response. And we deemed that students who scored 7 or above to be a successful student, and those who scored a 7 or 6 and below were deemed unsuccessful. So, using this criteria, we began to see patterns between successful and unsuccessful students. We found that successful and unsuccessful students were found at both levels here. Most students, successful and unsuccessful, did a reasonable job categorizing amino acids. And this is the phenylalanine is nonpolar, glutamate is negatively charged, and valine is nonpolar. In contrast, most unsuccessful students failed to make predictions. And these included the codes glutamate mutant would fold differently, would function differently, valine mutant would fold differently, would function differently. So, now that we see measures of success, we want to compare solutions between successful and unsuccessful students. To show the difference and similarities in solutions between experts, successful students, and unsuccessful students, a heat map of code prevalence was generated. And again, I'm going to explain this heat map. It's a little overwhelming. Starting from left to right, the left represents the type of knowledge, domain general, domain specific knowledge, followed by the more specific categories. And earlier we talked about the possibility of interactions between these types of knowledge. And this is where we found evidence that within our codes that there were interactions. And we included these here. And next to these were all the codes we use in our protein X problem. So following that includes the experts, the successful students, and the unsuccessful students. And I'll talk to you about our prevalence keys. So we use 4 levels of prevalence in the heat map, white being none, light gray being less than half, dark gray being more than half, and black being all of the population use that code. So this heat map shows all our codes, so you can see this is a good way to visualize all populations and compare all populations at once. And today I'm going to zoom in on 3 specific topics. student contradicting, non-polar interactions, and expressing tentativeness. And I'll start off with contradicting. This is a zoomed-in picture of the heat map. And contradicting is when a student stated a prediction but changed the prediction and changed their answer eventually. So this was mainly an unsuccessful, actually an only unsuccessful student code that you can see here. Successful students and experts were not coded for this. In addition, I'll talk about nonpolar interactions. Recognizing that nonpolar interactions occur between two nonpolar amino acids, many students got this wrong, but unsuccessful students especially struggled with this nonpolar interaction idea. Lastly, I'll talk about experts and successful students expressing tentativeness considerably when solving the problem, and we noticed that they displayed this tentativeness as a tool for problem solving to compare other scenarios before reaching the final conclusion. And this was characterized mostly by experts and successful students. So, to summarize, students and experts practice varying strategies when solving a problem. Both beginning and advanced students are found to be successful and unsuccessful. And compared to experts and successful students, unsuccessful students display difficulties that are targets for intervention. So, we talked earlier about our framework. We have specific and general, and including the three types of knowledge: declarative, procedural, and conditional. And Alexander predicted that there may be a correlation between these types of knowledge called interactions. And what we found is that, based on our coding and our data, we have found that these interactions do exist. And we have modified our model to represent that. So, we found that interactions exist between these categories. We see declarative procedural, declarative conditional, procedural conditional, declarative conditional procedural, combination of all three. So these are based on our qualitative data and results to support this emerging model. And in our work, we notice differences between experts and novices, which supports the literature. Also, successful students exhibit expert-like performances. Lastly, we found the three types of knowledge in our problem set. In our work, we've created a methodology for visualizing large bodies of qualitative data, and we hope that others can use this in other domains of biology, biochemistry for their research and their problem-solving inquiries. In addition, we identified that students struggle with biochemistry threshold concepts of the physical basis of interactions, more specifically with nonpolar interactions. And lastly, we have identified an emerging model of interactions and confirmed that these interactions do take place. So, in the future, we'll be testing our problems in a larger sample, and we hope to create instructional materials to target difficult biochemistry topics. In addition, we'd like to identify problem-solving strategies that lead to student understanding in biochemistry. So, with that, I'd like to acknowledge all our lab group, especially Dr. Lemons for all the guidance and mentorship, Cheryl and Stephanie and Jill, and also all the other undergraduate researchers in our project, Kush, Caitlin, Alex, Brian, and Morgan. Most of them can't be here today. We'd like to thank Bird UGA, the core collaborators group, and the threshold concepts in biochemistry group. Lastly, we'd like to thank the NFS for providing support and funding, and to the University of Georgia. Thank you.